It looks like we're ready to begin. Good morning. My name is Mike Leftashenko. I'm the Client Services Manager at New South Wales Health Statewide Biobank. I'm pleased to introduce the first of the three presentations we have for you. First, we hear from Alice Cochrane, Forensic Biologist in our Forensic Analytical Science Service. Before we start, I urge you to look in your attendee hub and look at the tab on the right-hand side. The Q&A tab is used for asking questions. I urge you to ask questions. We want to hear them. And so I'm handing over to you, Alice.
Lovely. Thank you, Michael. All righty. Thanks so much for having me today. And I'm really excited to be talking to you today about how we solve crime through family ties. Okay. But before we get into family ties, let's look at the big picture. What exactly does a forensic biologist do? The Forensic Biology DNA Laboratory is part of the Forensic Analytical Science Service, or FAS, at LIDCOM. And we perform biological and DNA testing on samples that originate from crime scenes across New South Wales. And these can be anything from property crime, such as break-enters or stolen motor vehicles, through to more serious crimes like drug matters, armed robberies, sexual assaults and murders, and everything in between. The larger portion of our samples only require DNA testing, and these samples are collected from the scene by the New South Wales Police and received by the laboratory in these tubes that can be loaded straight onto our robotic system. Sexual assault investigation kits make up the bulk of the rest of our crime scene work and are examined for biological fluids, which is mainly blood, semen and saliva, as well as DNA testing. And person reference samples are also processed by the laboratory. DNA profiles are uploaded to the criminal DNA database, which is managed by the laboratory, and any links to persons or other crime scenes are provided as intelligence to the police. If a matter proceeds to court, we can provide an expert statement and often give expert witness testimony. And when we talk about DNA, this is what we do. We extract DNA out of the substrate. We measure how much DNA is in the sample, amplify the specific areas we are interested in using a multiplex PCR kit, and separate and detect the amplified DNA using capillary electrophoresis. The DNA testing we do targets short tandem repeat areas, which vary between individuals. And it's this variation that makes each individual's DNA profile unique, except for in the case of twins. Today, I'll be talking to you about our autosomal DNA profiles, which targets multiple STRs across different chromosomes and is the main type of DNA testing carried out by the laboratory. But I'll also touch on our male DNA testing, which targets multiple areas on the Y chromosome only and is used in selected circumstances. The purpose of a DNA database search is to identify the donor of an unknown profile left at a crime scene. The unknown profile is uploaded and searched against the New South Wales and national DNA databases, where we're aiming to find a hit or valid match, ideally to a person sample, but links to another scene can also provide valuable information to an investigation. For the most part, unknown profiles undergo direct searching, where we are looking for the same or matching profile. But we can also do familial searching, which is a deliberate investigation of a DNA database in search of a first-degree relative of the unknown profile. In a familial search, we are looking for a similar profile. Close biological relatives share more DNA with each other than with a random person in the population. And familial searching uses the shared DNA to find a potential relative of the unknown profile. YSTR profiles also undergo direct searching on a database where once again, we are looking for the same profile. However, because YSTRs are conserved between paternal relatives, a YSTR link to a person, when taken in isolation, indicates that that person may be the donor of the crime scene profile. But equally, it could have been left by the person's father, brother, uncle, son, cousin, nephew, anyone on the paternal line. Thus, a YSTR link is both a direct and a familial link. So when we look at a family tree, we are expecting our familial search to identify the relatives shown in this green box. So parents, children, and siblings. But remember, paternally related males will share the same YSTR profile. So a YSTR link will pick up the close paternal relatives, but also those more distant paternal relatives that may be missed by the familial search. So now I've got a little bit of familial searching history for you. We started in 2013 with a pilot study of 5 serious crime cases. And for the next few years, a small number of familial searches were conducted, mostly on cold cases. In 2019, the focus turned to the unknown deceased and missing person casework that Carol will be covering in detail in the upcoming plenary session. So make sure you catch that one. The other thing that happened in 2019 was that the New South Wales Police formed a team dedicated to the authorisation and communication of familial search results for crime index samples. And this resulted in an explosion of familial search requests for crime index samples, as you can see in the graph in the top corner. So we had been doing familial searching using an in-house program, which allows for a point-in-time search. But we know that new person samples are added to the database daily, so we were keen to have familial searches running in a continuous fashion to search the expanding database. Luckily, the National DNA Database has a sister program called NIFA, which facilitates continuous familial searching. So we now use that routinely in conjunction with our in-house program. YSTR database searching is also a relatively new investigative tool in our laboratory. Although we validated our first YSTR kit around 15 years ago, these profiles could only be used for in-case comparisons, as there was no searching database that we could use to look for links in Australia. And this is still a really powerful tool in cases where autosomal DNA testing did not deliver a male profile suitable for comparison due to an overabundance of female DNA. In 2018, a new YSTR kit was validated, which tests more areas of the Y chromosome, including some rapidly mutating areas, which allows for greater discriminatory power. So in 2019, the lab began to upload casework and reference profiles to NIFA for national YSTR database searching, and we had our first YSTR link reported in 2020. So when we have an unknown profile from a crime scene that the police believe may have been deposited by a particular individual, that is a known POI, and that person does not have a reference on the DNA database, but they have a close relative who does have a reference on the DNA database, police can send through a familial comparison request. For these requests, police nominate a person and the relationship for comparison to the unknown profile. In the 3 years since this initiative has been implemented, we've had 187 familial comparison requests. And you can see that when police have nominated a parent-child or sibling relationship, that relationship is confirmed at least half of the time. When we have an unknown crime scene profile and the police have no POI for the matter, a familial search may be conducted. So the New South Wales Police familial searching policy dictates the sort of offences that familial searching may be used for. And it's mainly limited to serious offences such as homicides, sexual assaults and armed robberies, but it is used on both contemporary and cold cases. So when we have an unknown profile of sufficient quality in an appropriate case and it does not get a direct person link on the New South Wales or national databases, police send through authorisation for a familial search. And we use the autosomal and YSTR searching of the New South Wales and national databases to look for a relative of the unknown profile. And if a candidate is identified, this is reported to New South Wales Police. So familial searching is quite resource intensive compared to our routine laboratory work. And so it's only carried out on a tiny fraction of the samples processed by the lab. But in terms of the forensic labs around Australia, we're really leading the way in this space. So for the 432 crime index samples searched to date, we have provided 50 formal reports for familial candidates with a really high statistic and matching YSTR for males. And as a side note, this includes 73 priority samples for which we've provided 7 formal candidate reports. So taken together with the national familial candidates provided, this roughly equates to nearly a quarter of the searches, resulting in new leads for police to investigate. And to date, there have been 39 unknown profiles resolved thanks to information obtained from a familial search, which is nearly 80% of cases where a formal familial report has been provided. So YSTRs are primarily used in sexual assault cases, as well as for our familial searches. On the YSTR searching front, we are also leading the way in the forensic biology community in Australia, being the first jurisdiction to really take advantage of the national DNA database searching capacity. We've got about 2,000 crime samples and 12,000 reference samples searching on the Y database. Now, to give a little context, we've got over 150,000 reference samples on the autosomal database. So the Y database only has a fraction of the capacity that we'd like to have in an ideal world. But despite this more limited capacity, we have had over 1,400 Y links, but importantly, 107 of those have been what we call cold links to new individuals, providing new leads for the police to investigate. And as a result, 16 unknown profiles have been resolved. So I think you'll agree with me that these new familial searching options provide quite a valuable source of investigative leads in cases where other avenues have been exhausted. So I'd like to round out my talk today with a couple of case studies. In 2003-2004, a series of 13 sexual assaults occurred across Sydney's inner west, and these were linked by MO. Some of these were also linked by the DNA of an unknown male individual. Now, we did not get a direct person link from the database, and nor did we get a familial candidate when the profile was searched as part of the pilot study. But finally, in 2021, we had a breakthrough with a YSTR link being reported to the New South Wales Police. Now, interestingly, this link occurred because of a sample that had YSTR testing done as a result of a familial search for a different case. But the autosomal profile of the reference sample did not match the crime scene, so the story doesn't quite end there. The police needed to investigate the candidate's family tree and collected several covert samples from potential persons of interest before landing on the person whose DNA profile matched the profile found from these assaults. And this person has been arrested and is awaiting trial. Now, it turns out that the YCR link that provided the breakthrough in this case was from a cousin via a half sibling. So really quite a distant relationship that would never have been picked up in our familial search. And the second case I'd like to share with you is a 2008 murder where the victim was found in their own home. The cause of death was severe head trauma and loss of blood caused by sharp and blunt force applied to the face and head. An unknown male DNA profile was recovered from the murder weapon, the victim, and items from the scene, and was believed to originate from the murderer. But despite having this ideal DNA profile, no direct person link was obtained. Now, an enormous amount of resources were allocated by police and FAS over the years to identify the donor of this unknown profile, including the collection of over 70 reference samples. But unfortunately, months turned into years and still no direct link was found. Fast forward 10 years or so to 2020 and a familial search was authorised and a candidate was identified, finally a lead. Police investigated the family tree and obtained a reference sample from an individual that matched the unknown profile found at the crime scene. And again, this person has been arrested and is awaiting trial. So these cases are just two of many where a familial relationship proved to be the key to identifying the donor of the DNA left at the crime scene. Thanks so much for your attention, and I hope I've given you a little insight into how we use family ties to solve crime. And I'm happy to take any questions.
Thank you, Alice. It was an incredibly interesting talk, and the case studies were very sobering, especially seeing a map of your hometown, which are a backdrop to these case studies. If anybody has any questions, I encourage you to ask a question using the Q&A tab. It's on the right-hand side of the hub that you're all logged into. I'll give a few seconds for some to trickle in. Well, it's just a slow morning, Alice. It's early, and because it's such a sobering topic, none have filtered through. I'll just give it one more second. No, none have come up. Thank you again. Still none. All right. All right. The next person we have is a postdoctoral research fellow. It's Ebby Sim from the University of Sydney. Thank you, Ebby. I'll hand over to you now.
Thank you for the kind introduction, Michael, and good morning, everyone. I'm Ebby. I'm a postdoc at the University of Sydney, but I'm actually based at the ICPMR at Westmead Hospital. And today I'll be talking about how you can actually use genomics to actually survey a group of really interesting pathogens called Shiga toxin-producing Escherichia coli. And before I go into, oops, it's not changing. So this talk will just kind of be an amalgamation of an ongoing journey, which started off as a research project, trying to figure out what we can actually use genomics to kind of value add STEC surveillance and also to kind of provide a nice little report up to New South Wales Health Protection so we can actually have ongoing surveillance of this particular pathogen. As you can see here from this particular timeline, pre-2017, most of the testing were done by your usual methods of molecular testing and a bit of culturing. And then the earliest genome that we ever had a sequence of started about December 2017. And then in January 2020, that's when the research really started, or retrospective genomic analyses from genomes from 2017 all the way to mid-2020. And then we started to just kind of interrogate the genome, look at what key things will actually be of interest to our stakeholders at New South Wales Health Protection New South Wales. And as you can see that there's always an ongoing discussions with health protection, and also genomic. This research has been published, but we also performing ongoing surveillance. So just to give a quick introduction into this particular pathogen and why it's really exciting, exciting to me anyway, will be that Shiga toxin producing E. coli, or STEC for short, it's just basically a catch-all phrase for all pathogenic E. coli that actually produce a specific toxin called Shiga toxin. As you can see here from this particular cartoon, this ability is conferred upon E. coli by an infection of a bacterial virus known as the STX phage. So this particular STX phage, and I hope you can actually see my mouse cursor over here, in addition to carrying the phage DNA, it also carries the genes required to produce this particular toxin. And when it infects the susceptible E. coli, it can actually integrate into the genome of this particular bug, and then just gets vertically inherited. And when E. coli actually replicates, the bacterial phage, now known as a prophage, will actually replicate in concert with the bacterial chromosome. However, in events of stress, like say, for example, UV exposure, antibiotic treatments, this STX prophage can actually sense the distress signal that the particular bacteria is actually producing, and then will go through a process of induction where it will actually start to go on to a lytic life cycle to replicate its genome and generate viral progeny at the expense of its host. But at the same time, this also actually allows for the increase of Shiga toxin production. So this is why antibiotic treatment is not really recommended for any patients that it has a suspected STEC infection. So infections with STEC tends to have a range of clinical manifestations. It can either be an asymptomatic infection, but then again, for more clinical cases, they tend to present with acute gastroenteritis. And in a very small subset of patients, it can actually progress to a more severe disease state known as hemolytic uremic syndrome, or HUS for short. And both STEC and HUS are notifiable diseases, and there is a case definition, particularly for STEC, I've listed on this particular slide over here. So you either need an isolation of an STEC from feces, or the detection of the genes associated with the production of Shiga toxin, which is the STIX genes, by E. coli by PCR, either on an isolate or directly from clinical specimen. So there are two prominent virulence factors of STEC. I've already introduced one, and again, it's its namesake virulence factor called Shiga toxin. So STECs produce two types of Shiga toxin, STX1 and STX2, but this can actually also be further subtyped into minor subtypes. So for STX1, you can actually subtype to 1A, 1C, and 1D, or STX2, you can see that you can actually subtype it to a greater degree. And I'll get into this later, but the different subtypes actually do have ramifications for the potential to progress to severe disease states. The next prominent virulence factor of STEC is the locus of heterocyte effacement. So what this particular genomic feature is it actually encodes a molecular syringe that the E. coli can actually use to pump effector proteins into the host intestinal epithelial cells. And these effector proteins can kind of rewire the entire machinery of the particular cell to make it rather suitable for how the infection is going to happen. So for this particular molecular syringe known as a type 3 secretion system, what it can do is remodel the cell surface to actually have a nice little actin pedestal. So actins all gets polymerized, and then you have this really nice intimate adhesion between the bug and the particular cell. So it just kind of sticks on, and you don't really remove the bug from any shear forces whatsoever. And of course, not all STECs are really created equal, as I've alluded to from the previous slides. So some subtypes are actually more prone to actually, or rather have the potential to lead to more severe infections. So STX2A, 2C, and 2D, these are the three main subtypes that can actually increase the risk of development of HUS, and also the presence of multiple isogenic copies of these STIC genes as well. So let's say, for example, you have an E. coli with, let's say, 2 copies of the STX2A gene within it. That does actually mean that it can actually produce even more toxin than a particular STEC with 1. So that's a risk factor that we really want to interrogate. And the next thing will also be the presence of the lead or other key adhesion virulence factors, because to establish an infection, you actually need to adhere to the site on the human cells. So without key adhesion virulence factors, you can actually have STECs that are less virulent than others. And so noting these, there's actually a risk level designation designated by the FAO and the WHO, which kind of assess STECs into 5 different risk levels, risk level 1 through to 5, where risk level 1 tend to be the STECs of higher potential of causing severe disease, while risk level 5 will be other STECs that are more just probably cause diarrhea, and there'll be lesser risk to progress to HUS. So as you can see here, the key virulence factors that they actually target are the STIX genes as well. So just one thing to keep in mind before I go on to the other slides is that these risk factors will also be presented later, and then this is a key metrics that we actually look at for ongoing STEC surveillance. So like I've mentioned, the retrospective research really started on 2022, and then with the first genome in December 2017. But there are workflows already present for interrogating STECs that actually isolate from patients in New South Wales. So I've introduced the laboratory evidence in the previous slide, and also the different risk levels that you can actually classify STECs into. And this is the classical workflow prior to whole genome sequencing, rather through the introduction of whole genome sequencing in ICP-MR. So fecal samples will be submitted to a diagnostic microbiology laboratory where PCR will be performed directly on the clinical specimen. And then once it's actually pings positive for either STIX1, STIX2, or both STIX1 and STIX2, culture will be attempted. So unfortunately, culturing of STEC is a non-trivial task because you're kind of pulling out a particular E. coli from a soup of E. coli that's actually present within the fecal sample. So the capture rate of what we've assessed so far is about 33% of the samples will result in isolate, while we actually lose about more than 70% of the other STECs. But however, this will actually fit the case definition, and it can be reported that patient actually has an STEC infection. So for the isolates that can be isolated from fecal samples, it'll be forwarded to the New South Wales Enterics Reference Laboratory for a bit of O serotyping. So O serotyping is where we actually target the somatic antigen of this particular bacteria. And then this is a classical way of how we actually describe said pathogen. So if you would have listened, kept an eye on the news, things like E. coli 0157, H7. So this is a key, a very prominent STEC serotype, and this is how we kind of report this. And also, once we can isolate, the Enterics Reference Laboratory will also bank them as a stock so that we can actually go back to it if we need to. So with this current workflow, definitely we can meet the laboratory evidence of case definition. But again, for reasons of surveillance, it's not really ideal because you actually lose a lot of information by just targeting a PCR on fecal sample or an isolate without any other thing else. So while it can be used to monitor the epidemiology of said infection, it's rather hard. And of course, to have a nice surveillance system where you can kind of put STECs into different risk levels, PCR is not sensitive enough to actually break them into the different subtypes. So we actually do not have a very good hold on the type of, or rather what risky STECs are actually circulating in the population. And this is where whole genome sequencing really comes in. So we've added additional workflow for all isolates that can actually be isolated from clinical samples. These plates will be forwarded to the Microbial Genomics Reference Laboratory at ICP-MR, and then the team will actually extract DNA from these isolates and sequence it on the Illumina NextSeq platform, which is a short-read sequencing platform. And then once we get the sequencing data, there'll be 2 different tiers of analyses that will be done. So the first will be a classical typing and virulence gene detection, while the second type of analysis will be genomic clustering to find any genomic similarities between the strains. So the first tier on genomic typing and virulence gene detection is just focusing on the key markers that are traditionally used to interrogate STECs. So things like serotyping for the O and H antigen, MLST and multi-locus sequence typing, detecting any AMR genes actually present within the genome, and as well as a full interrogation of what other virulence factors that this particular STEC carries. So one of the things that we kind of did in ICP-MR is the development of a STEC virulence barcode to actually capture key pertinent information that could be reported up, and also to actually track said STECs that's actually being sequenced, and also hopefully get a sense of the epidemiological trends in New South Wales. So this barcode actually allows for the inference of multiple isogenic STX genes present within the genome of the STEC, and as well as subtyping of your key STEC genes, which is both your intimin and as well as your STX genes. And from this, we can actually assign a risk level 1 through 5 as demarcated by the FAO and the WHO. So in this particular slide, I'm just showing the extent of the serotypes and the virulence barcodes we actually seen in the STECs that's circulating in New South Wales. So the virulence barcode is not always stratified to the serotype that we actually sample. And as you can see in this particular diagram, the Sankey plot, on the left-hand side, it's the STEC serotype, and on the right-hand side is the virulence barcode, and the gray connect line is basically a relationship between serotype and the virulence barcode. So as you can see, if we take, for example, 0157H7, which is our most prominent STECs that we actually isolate and sequence, you can see, again, not all 0157s actually made equal, because if we look over to the right-hand side of this particular diagram, you can see that it actually has virulence genes that either place it in a risk level 1 or as tame as a risk level 4. So again, not all STECs are created equal. And this is really due to basically one thing that we don't really track very well in this time and space, which is the STX profiles that are really circulating around in New South Wales and also globally. And with all this data, we can actually start to look at trends that will be very useful for New South Wales health protection, and also to have ongoing surveillance of this particular bug. So what we can see here in this particular diagram, or this particular line plot, is basically the proportion of sequence STECs belonging to this different risk levels, risk level categories from 1 through to 5. And we can see that we actually do have a higher number of risk level 3 and risk level 4 STECs, with very low numbers of risk level 1 and risk level 2. And this also really corroborated with Australia being a really low incidence country. And I guess Australia is kind of blessed with STECs that don't particularly carry the more virulent toxin gene subtypes as compared to other international our international counterparts. So with genomics, we can not only look at the virulence factors, but we can also start to see if we can actually cluster the genomes together and see how similar they are. And this can actually inform things like transmission chains, or rather to see what's actually circulating around in the background. So we have two different analyses that will be performed. The first is basically a whole genome MLST, where we target 2,513 genes, and then we just pump it through the software, and this will actually give a very broad resolution assessment of genomic similarity. So as you can see here, it's always presented in a minimum spanning tree diagram, where each individual nodes represent a particular isolate, and the edges separating them is basically the number of allelic difference. So the smaller the line, the more related to the more related the STECs are, the more distantly related they are. So as you can see here, it all really clusters really nicely together, with all the serotypes really forming a tight little cluster. Excuse me. And then the second resolution that we actually go to will be interrogation of core SNPs. So this is a more finer resolution of clustering based on mapping all your reads that you get off your sequencer onto a reference genome. And then we identify the core SNPs in your entire data set and generate a little matrix, which can be used to generate a tree, somewhat like this. And we can start to see, or rather interrogate genomic similarity or differences. So over here in this particular slide is a tree of the current 0157s, H7s that we actually have. Good thing is most of them are really sporadic and really distinct from each other. But again, we do have the capabilities of actually pinpointing any transmission chains, and or rather inform transmission chains and interrogate of genomic similarity. And of course, one key thing, again, for which kind of leads to future research in this point, will be the development of reference genomes to actually map these sequencing reads onto. So there is a strict criteria because you don't really want to map reads from an STEC that's an 026 to a STEC that's 0157 because they are genomically distinct, as you can see from the previous, as you probably would have seen from the previous slide. So we need the same serotype and MLST. And of course, Australia being a geographical bottleneck, there seems to be a little bit of unique population circulating around the STEC. So STECs isolated from Australia tend to be a bit more distinct from what you actually isolate from the rest of the world. So if we actually have a reference genome from Australia, that will pretty much be a very high criteria. And as well as most genomes up to now are generated via short read sequencing, which you can't really resolve the entire genome of a particular bacteria. So any genome that actually has been resolved by long read sequencing is also ideal to be used as a reference genome as well. So we've presented 3 different workflows over time, and what you can see that whole genome sequencing or actually having genomic surveillance really value adds the process that we can actually submit to stakeholders, where not only can we detect the STX genes, but we can start to do things like assessment of how related these particular isolates are to each other, and as well as providing a genomic evidence that can either support or refute transmission links. So in summary, the present methods of country-independent testing and country-without sequencing can mask additional information which could be informative for surveillance, and that we, the team at ICPMR, we developed an STEC virulence barcode, and we've also published this, and we hopefully that this actually gains traction. Either locally it is reported to health protection, but also internationally to see if this actually gains a nice little way that we can track the virulence of STECs. And also within our current data set, so far, majority of sequence STECs fall into the lower risk groups from group 3 to group through to group 5. And of course, 0157, H7 dominated our data set. And I also mentioned that this is an ongoing thing. So our future direction on what we have a little bit of funding, thanks to Sydney ID, is to actually utilize long read sequencing to resolve key STEC serotypes that we don't really have a complete genome for. And this would be really, really helpful for us to see how different our STECs in Australia are, or if there's actually other genomic features that will be really key to interrogate for a more optimal surveillance system. And with that, I'd like to thank everyone from the Microbial Genomics Reference Laboratory at the ICPMR, the New South Wales Enterics Reference Laboratory, also housed in the ICPMR, and as well as all diagnostic microbiology laboratories in New South Wales. So I've come to the end of my talk, and I'm happy to take any questions.
Thank you, Abby, especially the great detail that you included in your talk. We don't have any questions yet, but you did go a little bit over time, so we went into bonus time here. Just a quick question. As a parent, what are your best tips to avoid infection? You know, I'm always surprised we have sort of low incidence, given that we've got really hot summers and we all like our steaks medium rare. I mean, if you've got a take-home tip for us, what would you suggest be the best habit to adopt?
Well, best habit will be making sure that meat is definitely cooked, and salads, give them a good wash before consuming salads as well, because that could be a vector for infection. And the key thing that I guess we've learned from the pandemic: wash your hands, wash your hands, wash your hands. Very good at preventing enteric infections. Excellent advice.
Thank you, Abby. Before we go to our next speaker, I just want to encourage you to take note that we have a tab on the right-hand side of the of your screen for polling. If you click on it, you'll be able to vote for your favourite speakers this session. And again, for our next speaker, please, I encourage you to ask questions during the talk. Thank you again, Abby. And now for our last ECR presentation, Dr. Shona Chandra, postdoctoral research fellow from the New South Wales Health Pathology ICPMR at Westmead. I'll hand over to you now, Shona. Okay.
Hi, sorry.
hear you loud and
Can
clear,
you guys
Shona.
see and hear me? I'm a bit
Yes,
late.
thank you.
Okay, so thank you, Michael, for the introduction, and thank you to Evie and Alice for the really interesting presentations before me as well. Oh no. Okay. So in the spirit of reconciliation, I just wish to start by acknowledging the traditional custodians of country throughout Australia and their connections to land, sea, and community. Today, I am on the lands of the Darug people, and I wish to pay my respects to elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. So today I'm just going to quickly talk about Streptococcus pneumoniae, also known as the pneumococcus. So strep pneumoniae is a gram-positive facultative anaerobic bacterium, which is commonly found as part of the normal flora in the upper respiratory tracts of healthy individuals. But in immunologically naive persons, such as children under 5 years of age and adults over 65, the pneumococcus can become pathogenic. There are at least 100 known serotypes associated with Streptococcus pneumoniae. However, only a handful of serotypes cause the majority of disease. Streptococcus pneumoniae can cause a whole host of different illnesses of varying severity, depending on which part of the body is infected, and it can be non-invasive or invasive. Invasive pneumococcal disease occurs when the pneumococcus migrates into typically sterile sites such as blood or CSF. Compared to other pathogenic pneumococci, serotype 3 resists antibody-mediated clearance, has a high invasive capacity, and causes severe infections with high mortality rates. Serotype 3 is a common cause of invasive pneumococcal disease in adults, and the introduction of the pneumococcal conjugate vaccine has dramatically decreased the incidence of IPD in children. So globally, the introduction of the heptavalent vaccine, the PCV7, has resulted in a substantial reduction of IPD. However, soon after, an increase in IPD due to non-vaccine serotypes was observed, prompting the development of expanded serotype conjugate vaccines. So currently, we also have a 13-valent conjugate vaccine, which contains 6 additional serotypes beyond those in PCV7. So these are the ones that PCV7 had, and this is PCV13. So all of PCV7 plus these additional 6, including serotype 3. In Australia, invasive pneumococcal disease has been a nationally notifiable disease in all states and territories since 2001, because it's a leading cause of morbidity and mortality among children, elderly, and Aboriginal and Torres Strait Islander peoples. Although a marked decline in IPD cases was observed from 2005, when the PCV7 was introduced to our national immunisation program, case numbers of non-vaccine type disease, including that of serotype 3 at the time, started to steadily increase until 2011, when the PCV13 formulation replaced the PCV7 in our routine immunisation schedule. While the introduction of PCV13 in accordance with the 3+0 schedule initially appeared to control cases of serotype 3 cases in Australia, they eventually started to increase again. So this is just the vaccination schedule that's recommended for all children less than 5 years of age in Australia. So the pneumococcal conjugate vaccine, or PCV, is provided free of charge under the National Immunisation Program. So children identified as most at risk for invasive pneumococcal disease are recommended a more extensive schedule. So this one down here. And it's recommended that they receive the 13-valent PCV conjugate, so the conjugate vaccine, in addition to a 23-valent pneumococcal polysaccharide vaccine. So the 23-valent PPV is funded for children from 2 years of age and also adults with medical conditions that increases their risk of invasive pneumococcal disease and is listed on the pharmaceutical schedule. So these vaccines are really designed to protect people from severe forms of of the disease, such as bacteremia, meningitis, and death. So we have a vaccine which covers serotype 3. It's publicly funded in Australia and it's made available for all children in Australia. So how has serotype 3 persisted and resisted antibody-mediated clearance despite its inclusion in the PCV13 formulation? Which then leads us to the aim of this study, which was to understand the evolutionary and genomic characteristics of invasive serotype 3 disease, which continues to persist despite our high vaccine coverage. So this was a retrospective longitudinal study of reported serotype 3 disease cases in New South Wales. So what basically happens for us to get these isolates is person falls sick and then they present to a doctor, and if they're then referred to a diagnostic pathology for confirmatory, or rather they're referred to a diagnostic pathology, and if they are found to have Streptococcus pneumoniae, they are then, that sample is then referred to the New South Wales Pneumococcal Reference Lab, which is based at Westmead Hospital, for serotyping. So if it's identified as serotype 3, we then perform genomic sequencing on it, and then we analyze the results. So the New South Wales Pneumococcal Reference Lab is the reference lab for the invasive pneumococcal disease surveillance. And so what they do is they serotype all pneumococci isolates referred from private and public diagnostic pathology labs in New South Wales and the ACT. So first we'll do serotyping by the gold standard quelling test reaction. So it looks at the cells and whether or not they swell. And that test reaction uses pool type and factor specific antiserum. So for this study, we used all historical serotype 3 IPD isolates referred to the New South Wales Pneumococcal Reference Lab. So there was 54 isolates collected from children under 5 years of age, and we tried to match the number of isolates collected each year from adults. So we got about 81 from adults, and these were collected between September 1999 and December 2018. Then we performed bioinformatics analysis. And with our quality filtering, we got rid of 10 samples from the analysis, but we also included 182 international genomes from 2 different studies just to compare how we're doing. So in the pre-PCV era, serotype 3 colonization rates and incidences in the pediatric population were relatively low. So this is when PCV7 was introduced, and this is serotype 3 cases in children in New South Wales. However, studies have shown several cases of pneumonia complicated with empyema, pleural effusion, or both produced by serotype 3 in vaccinated children. We started to see an increase in non-vaccine serotypes following the implementation of PCV7. And although a marked decline in IPD cases was observed from 2005 when PCV7 was introduced, case numbers of serotype 3 IPD started to steadily increase until the PCV13 replaced the PCV7 in the routine immunization schedule. While the introduction of PCV13 in accordance with the 3+0 schedule initially appeared to control serotype 3 IPD cases in Australia, so we can see the case numbers do start to drop off a bit. Case numbers started to increase again until the PCV13 immunization schedule was amended to a 2+1 schedule. While the initial vaccination scheduling could account for the high representation of invasive serotype 3 disease cases observed in Australia, there is not enough supporting evidence to say that it's actually the vaccination schedule. So this column graph here just demonstrates the incidence of serotype 3 disease cases in children. So that's the blue bar, and adults, which is the red bar for New South Wales. And we have the overlaid line graph in black showing the total number of invasive pneumococcal disease cases in Australia on the primary axis. The filled triangles here mark the years that the vaccines were introduced. And in 2011, the PCV13 was administered with the 3+0 schedule, as I said before, but then in 2018, due to the uptake of cases in children, it was then amended to a 2+1 schedule. However, since 2011, a 3+1 schedule is recommended for identified higher risk groups of children as well. So this is a maximum likelihood phylogeny of the assembled genomes from serotype 3 invasive pneumococcal disease cases, which was constructed. So this tree was constructed using ggtree, and this tree was constructed on microreact. So this circular tree and this unrooted tree is actually the same tree, but I've just collapsed the novel clade in the circular tree. So for illustrative purposes, this novel clade in this unrooted tree is actually scaled down to 5%, and because it throws out, it's quite divergent. So as you can see, there's SNP difference. The minimum SNP difference is 8,500 SNPs. So because it's quite divergent, I've just condensed it a bit for this tree and completely hidden it for this tree. So the global persistence of serotype 3 is likely attributed to this recently emerged clade 2 that we can see here in light blue. And while there's a higher prevalence of antimicrobial resistance among clade 2 globally, we haven't yet observed that in Australia. So the Australian genomes here are denoted by this yellow bar on the outside, and AMR is denoted by the grey bars. So we don't actually have much overlap with AMR and our Australian isolates, which is very good. So among all of our serotype 3 isolates from New South Wales, we only found inferred antibiotic resistance in 5 genomes, which is these 5 here. And what percentage of overseas clade 2 is resistant? Well, 20 samples of the 20 samples of 71 in clade 2 subset had inferred resistance for international genomes, whereas only 5%, so one sample had it of, so this is the TET-M resistance in Australia. So overall, I think we're doing quite well. But what is interesting to note is that in the pre-PCV7 era, we had more novel clades than in the PCV7 era, and since adopting the PCV13 formulation, we're starting to see an increase in novel clades as well. And we only ever saw the emergence of clade 2 in our Australian genomes following the PCV7 use. So with the huge increase in bacterial and genomic sequence data available because of next-gen sequencing, they have assumed that there is, or international studies have assumed that there's this link between serotype 3 clade 2 and antimicrobial resistance due to the acquisition of this TN916-like transposon. And many of the transposons in this family are capable of excision, integration, and conjugation. And nearly all of the TN916-like elements discovered to date encode tetracycline resistance. However, increasingly, it's becoming resistant to many other antimicrobials as well. So the international pneumococcal serotype 3 genomics data have reported this association. However, this is not yet the case for Australia, and in our data, we've not yet observed a link between AMR and clade 2, and we have not observed this TN916-like transposon in our data set. So what does this mean for us? So the global persistence of serotype 3 is likely attributed to this recently emerged lineage clade 2 within our samples, our serotype 3 samples. And while there is a higher prevalence of antimicrobial resistance among clade 2 globally, we haven't yet seen that in our serotype 3 isolates, and hopefully it stays this way. So while the initial vaccination scheduling could account for the higher representation of invasive serotype 3 disease cases in Australia, there is not yet enough supporting evidence to indicate this. Just like to acknowledge and thank all of these people, namely Miss Bianca Crowder and Dr. Shaheen Aftade from the Pneumococcal Reference Lab. This study really wouldn't have been possible without them and their isolates, as well as the Microbial Genomics Reference Lab, who really helped with the genomic sequencing of this, Sydney Informatics Hub for providing us with access to the HPC to facilitate the running of the analysis, as well as the National Notifiable Disease Surveillance System for providing us with the data that we've also used in this. And if anyone has any questions, they can feel free to email me as well. Thank you.
Thank you, Shona. That was very detailed. I think you've continued the trend across all of the 3 presentations. I think the detail was so thorough. That's why we didn't have questions. Well done. I just want to encourage everybody to use the polling tab to vote for your favourite presentation. There's still no questions. So Shona, there are no questions. I apologise, but I encourage you all to join the next session, which is the first plenary session of our forum. It's only just begun, so that if you transfer to this option in the next few minutes, you won't miss too much of a talk. Again, thank you to all the speakers, and enjoy the rest of the forum.